Hello dear students, welcome to a lecture on mechanism of enzyme action. In today's deliberation we shall learn why enzymes have higher catalytic power. What is the importance of enzyme substrate complex in enzyme catalyzed reactions? We will find out the relationship between activation energy and reaction rate. We will also discuss that lowering of activation energy is the basis of rate enhancements in enzyme catalyzed reactions. Finally, we will study the various mechanisms of lowering of activation energy. First of all, let me give a brief introduction about enzymes and how they differ from other catalysts. Enzymes are the catalysts of biological origin, which accelerate the various biochemical reactions in biological systems. Almost every biochemical reaction is catalyzed by an enzyme. Like all other catalysts, enzymes increase the rate of chemical reactions without themselves being consumed or permanently altered, and without altering the chemical equilibrium between reactants and products. However, they differ from ordinary chemical catalysts in higher catalytic power, that is, higher reaction rates, greater reaction specificity, milder reaction conditions, and capacity for regulation. Few non-biological catalysts have all these properties. However, the catalytic mechanism employed by enzymes are identical to those used by chemical catalysts. Enzymes are simply better designed. Under biologically relevant conditions, uncatalyzed reactions tend to be slow because of many problems. An enzyme circumvents these problems by providing a specific environment within which a given reaction can occur more rapidly. This they do by forming enzyme substrate complex and by lowering of activation energy. Now let us first study formation of enzyme substrate complex. The catalytic activity of enzymes involves the binding of their substrates to form an enzyme substrate complex. The substrate binds to a specific region of the enzyme called the active site. The binding is a very specific interaction. Once a substrate is bound to the active site of an enzyme, multiple mechanisms can accelerate its conversion to the product of the reaction. While bound to the active site, the substrate is converted into the product of the reaction, which is then released from the enzyme. The simple enzyme catalyzed reaction can thus be written as E plus S gives ES gives EP, which in turn gives E and P, where E indicates the enzyme, S substrate, ES indicates enzyme substrate complex, EP indicates enzyme product complex, and P indicates the product. The enzyme E appears unaltered on both sides of the equation. Enzymes, like other catalysts, do not affect reaction equilibria. They accelerate the reaction in both the directions. That is, the reaction reaches equilibrium much faster when the appropriate enzyme is present. The interactions between the enzyme and substrate in enzyme substrate complex promote the formation of the transition state in enzyme substrate complex, resulting in lowering of the activation energy. Now let us discuss lowering of activation energy, how activation energy is lowered. As per transition state theory, a chemical reaction of substrate S to form product P goes through a high energy unstable intermediate state called transition state at some point in the reaction. This transition state has a higher free energy than either substrate or product P, as shown by a transition state diagram. A transition state diagram or reaction coordinate diagram is a diagram showing the energy changes during the reaction. It is the plot of the free energy of the system against the progress of the reaction. which is also called the reaction coordinate. The starting point for either the forward or the reverse reaction is called the ground state. There is an energy barrier between substrate S or product P that must be overcome for the reaction to proceed in either direction. The top of the energy hill, that is the highest free energy state, is the transition state. Any transient chemical species that has a finite chemical lifetime is called reaction intermediate. For example, enzyme substrate complex or enzyme product complex. It is quite different from the transition state. In reaction coordinate diagram, the intermediates occupy valleys while transition state is denoted by a peak. The difference between the energy levels of the ground state and the transition state is called the Gibbs free energy of activation, or simply the activation energy. It is denoted by delta G double dagger. For forward reaction, activation energy is the difference in free energy between the substrate and the transition state, or we can write it delta G is equal to free energy of the transition state minus free energy of the substrate. Now, having learned activation energy, let us study the relationship between activation energy and the reaction rate. Activation energies are energy barriers to chemical reactions. The rate at which a molecule undergoes a particular reaction decreases as the activation barrier for that reaction increases. The rate or velocity of the reaction, which is denoted by V, is given by the expression V is equal to Ks, where K indicates the rate constant and S the substrate concentration. This equation can be also written as V is equal to K into T divided by H into substrate concentration exponential of minus delta G divided by RT, where K indicates Boltzmann's constant, H indicates Planck's constant, T temperature, S substrate concentration, and delta G indicates the free energy of activation, and RT, where R indicates the gas constant and T the temperature. As you will see in this equation, there is a negative exponential relationship between rate of a reaction and its activation energy. In other words, the rate of a reaction depends on this activation energy. The higher activation energy corresponds to a slower reaction. Hence, we can say that reaction rates can be increased by lowering the activation energies. The activation energy for the uncatalyzed reaction is the difference in energies of substrate and its transition state. Similarly, the activation energy in the catalyzed reactions is the energy difference between enzyme substrate complex and its transition state. As you will see in the diagram, the energy barrier, that is the activation energy between enzyme substrate and its transition state, is less than the energy barrier between substrate and its transition state. In terms of free energies of activation, we can say that delta G for catalyzed reaction is less than delta G for uncatalyzed reaction. That is, the activation energy for the catalyzed reaction is lower than the activation energy for uncatalyzed reaction. In other words, enzymes lower the activation energy. The lowering of activation energy is the difference between the values of delta G for the uncatalyzed reaction and catalyzed reaction. When the free energies of ground states of S and ES are equal, then lowering of activation energy is simply the difference between free energy of transition states of substrate and enzyme substrate complex. This lowering of activation energy indicates the efficiency of the catalyst and is related to the rate enhancement by the equation rate enhancement is equal to exponential of delta delta G or T, where delta delta G indicates the lowering of activation energy, R indicates gas constant, and T temperature. That is, rate enhancement occurs exponentially with the lowering of activation energy. Thus, enzymes enhance reaction rates by lowering the activation energy, that is delta G double dagger. The combination of substrate and enzyme creates a new reaction pathway whose transition state energy is lower than that of the reaction in the absence of enzyme. In other words, enzymes facilitate the rapid formation of the transition state. While reaction rates depend on activation energies, reaction equilibria, that is spontaneity, depends on free energy change for the reaction. Since enzymes lower only the activation energy without altering the free energy change of the reaction, they do not make non-spontaneous reactions to occur spontaneously. Now we will discuss the various mechanisms of lowering of activation energy. The enormous and highly selective rate enhancement and the dramatic lowering of activation energies for enzyme catalyzed reactions occur through following ways: non-covalent interactions between enzyme and substrate, rearrangement of covalent bonds during an enzyme catalyzed reaction. First of all, let us study non-covalent interactions between enzyme and substrate. A large number of non-covalent bonds and interactions such as hydrogen bonds and hydrophobic and ionic interactions are formed between substrate and enzyme in an enzyme-substrate complex. These interactions are accompanied by a release of free energy called the binding energy, which is denoted by delta Gb. This binding energy contributes to a specificity as well as to the catalysis. Much of the catalytic power of enzymes is ultimately derived from this binding energy, as it is a major source of free energy used by enzymes to lower the activation energies of reactions. As per equation, V is equal to KT divided by H into substrate concentration E minus delta G divided by RT. Or as per equation, rate enhancement is equal to exponential of delta G divided by RT. Delta G must be lowered by about 5.7 kilojoules per mole to accelerate a first order reaction by a factor of 10 under conditions commonly found in cells. The energy available from formation of a single weak interaction is generally estimated to be 4 to 30 kilojoules per mole. The overall binding energy available from a number of such interactions is therefore sufficient to lower activation energies by the 60 to 100 kilojoules per mole, which is enough required to explain the large rate enhancements observed for many enzymes. The same binding energy that provides energy for catalysis also gives an enzyme its specificity, that is, the ability to discriminate between a substrate and a competing molecule. Now let us study the mechanisms based on these non-covalent interactions, that is, the mechanisms involving binding energy. The lowering of activation energy is brought about by binding energy through following mechanisms: stabilization of transition state, entropy loss and destabilization of ES complex, proximity and orientation effects. Let us take these three one by one. First of all, we will take stabilization of transition state or preferential binding to transition state complex. We know that energy barrier between enzyme substrate complex and its transition state is less than the energy barrier between substrate and its transition state. This means that the enzyme must stabilize the transition state complex more than it stabilizes the enzyme substrate complex. Put it another way, enzymes are designed by nature to bind the transition state structure more tightly than the substrate or the product. In other words, enzyme active sites are complementary not to the substrate per se, but to the transition states through which substrates pass as they are converted to products during an enzymatic reaction. An enzyme completely complementary to its substrates would be a very poor enzyme, as it would stabilize the enzyme substrate complex, thereby decreasing its free energy. In a reaction coordinate diagram, this kind of enzyme substrate complex would correspond to an energy drop from which the substrate would have difficulty escaping. The result is an increase in the activation energy. Such an enzyme impedes the reaction and would be useless. When an enzyme is complementary to the reaction transition state, it means that optimal interactions between substrate and enzyme occur only in the transition state. The free energy, that is binding energy, released by the formation of these reactions or interactions partially offsets the energy required to reach the top of the energy hill. In other words, the transition state gets stabilized, that is, it has less free energy as compared to that in uncatalyzed reaction, thereby resulting in lower net activation energy. Now, let us study second mechanism based on binding energy, that is, entropy loss and destabilization of ES complex. The binding energy stabilizes both the ES complex as well as the transition state, but to different extents. If the ES complex and the transition state for the reaction are stabilized to equal extents, the catalysis does not occur. However, catalysis will occur if the transition state is stabilized to a greater extent than the ES complex. Since the binding energy stabilizes the enzyme substrate complex also, even if to lesser extent than the transition state complex of enzyme substrate, it deepens the energy well of the enzyme substrate complex, thereby increasing the difference in energies between enzyme substrate and its transition state. This results in lowering of the rate of the reaction. This effect of binding energy on stabilization of enzyme substrate complex needs to be compensated by some mechanism which raises free energy, that is, which makes the enzyme substrate complex to undergo destabilization. This is accomplished in two ways. Number one, loss of entropy due to the binding of substrate to the enzyme, and number two, destabilization of enzyme substrate complex by strain, distortion, dissolution or other similar effects. Now let us study the effect of loss of entropy. Binding of substrate to enzyme to form the ES complex leads to a net loss of entropy, that is T delta S. This reduction of entropy in turn increases the free energy of ES complex, as we know in thermodynamics. Second, destabilization of enzyme substrate complex. Destabilization of enzyme substrate complex can occur due to the structural strain or the distortion of the substrate due to their binding, due to dissolution of hydration shells from charged groups, due to electrostatic interactions between charges of like sign. Destabilization makes enzyme substrate complex less stable and more reactive, thereby raising the energy of enzyme substrate complex. Now, whether by strain, dissolution or electrostatic effects, this increase is summed in the term delta GD, the total energy of destabilization. The destabilization energy and the entropy loss on binding raise the free energy of enzyme substrate complex by the amount delta GD minus T delta S, while the binding energy delta GB lowers its free energy. As noted in this figure, the energy difference between substrate and enzyme substrate complex is delta GB minus delta GD minus T delta S. The transition state, on the other hand, is subject to no such destabilization, and the difference between the energies of transition states of substrate and enzyme substrate complex is essentially delta GB, the full binding energy. In other words, the energy of transition state of enzyme substrate complex is decreased more than that of ground state of enzyme substrate complex, thereby resulting in lowering of activation energy in enzyme catalyzed reactions. Now, the third mechanism, that is proximity and orientation effects. Chemical reactions go faster when the reactants are in proximity, that is near each other. Enzymes take the reactants out of dilute solutions and hold them close to each other or to catalytic residues. Enzymes not only bring substrates and catalytic groups close together, but they also orient them in a proper manner suitable for catalysis. Proximity and orientation effects may also lead to electrostatic interactions that result in the stabilization of transition state, which is sometimes also called electrostatic catalysis. Now let us study rearrangement of covalent bonds during an enzyme catalyzed reaction. In most enzymes, the binding energy used to form the enzyme substrate complex is just one of several contributors to the overall catalytic mechanism. Besides binding energy, catalysis also occurs by transient covalent interaction of enzyme with a substrate or group transfer to or from a substrate. These transient covalent interactions or group transfers lower the activation energy, thereby accelerating the reaction by providing an alternative lower energy reaction pathway. Now let us go through the mechanisms based on transient covalent interactions or group transfers. These include general acid-base catalysis, covalent catalysis, and metal ion catalysis. First of all, let us take general acid-base catalysis. Charged intermediates of many biochemical reactions are unstable and can often be stabilized by the transfer of protons to or from the substrate. Non-enzymatic reactions can involve either the constituents of water, that is H positive or OH negative, alone, or other weak proton donors or acceptors for the proton transfers. Being weak acids or bases, the intermediate is not effectively stabilized every time it forms, thus rendering the catalysis slow. However, the active sites of an enzyme contain a number of amino acid side chains that act as strong acids or bases, readily donating or accepting a proton from transition state, thereby stabilizing it and enhancing the rate. Now let us study covalent catalysis. In covalent catalysis, the active site contains a reactive group, usually a powerful nucleophile that forms a transient covalent bond between the enzyme and the substrate. This results in the new pathway that has lower activation energy than the uncatalyzed pathway. Now, metal ion catalysis. Nearly 1/3 of all known enzymes require one or more metal ions for catalytic activity. Metals can participate in catalysis in several ways. Number 1. Ionic interaction between an enzyme-bound metal and a substrate can help orient the substrate for reaction or stabilize charged reaction transition states. Number 2. Metals can also mediate oxidation and reduction reactions by reversible changes in the metal ion's oxidation state. Metals also act as electrophilic catalysts, stabilizing the increased electron density or negative charge that can develop during reactions. Fourth, metals also catalyze reactions by electrostatically stabilizing or shielding negative charges. These mechanisms, whether stabilization of transition state, entropy loss and destabilization of ES complex, proximity and orientation effects, general acid-base catalysis, covalent catalysis, or metal ion catalysis, are not mutually exclusive, and a given enzyme might employ a combination of several catalytic strategies in its overall mechanism of action to bring about a rate enhancement. For most enzymes, it is difficult to quantify their contribution of any one catalytic mechanism to the rate and or specificity of a particular enzyme catalyzed reaction. Now, the conclusions. Finally, we can conclude that enzymes have higher catalytic power because of the formation of enzyme substrate complexes that facilitate the rapid formation of the transition state and thus help in lowering of activation energy. The lower the activation energy, the exponentially higher the rate enhancement. The lowering of activation energy occurs through various mechanisms involving non-covalent as well as transient covalent interactions, among which the binding energy is the major source of lowering of activation energy. With this, we conclude today's lecture on mechanism of enzyme action. Thank you.